Thanks, and hopefully I'm not going to be just tying shoelaces here, but we'll see. But in the spirit of how did I become a marine biologist, as some of you might know, I'm half American and half Greek, and I spent a lot of time as a kid face down in the Aegean Sea with my snorkel and mask firmly adhered to my face. And actually my first experience with coral reefs was in some of the reefs right off of Cairns, and that was in 1991 when I was an exchange student studying at the University of Melbourne, and I fell in love with those reefs, and I said, I'm going to be back here someday, and so I'm really happy to be back here. So the project I'm going to talk about today is really exciting to me because it involved my whole lab group, and we got students involved in this, and it's been an interesting ride. But I wanted to, this is a global data set, and so the data that I'm going to be talking about today comes from many sea countries around the world. And so I just want to acknowledge that many traditional custodians will have firm attachments to these regions that are represented in what I'm going to be talking about. So I'm going to be talking about hidden dimensions of biodiversity, and I mean two things in particular. I mean cryptic species, which I'm defining as genetically distinct groups that are not easily recognizable just by human eye, and hybridization, which is when you have rare mating between species leading to gene flow between them. And so just to give you an example of how this can be, this is some data that came out of my group recently. This is Stylophora pistillata from the Great Barrier Reef, and this actually, in this picture, are 3 different cryptic species, and I'll give you the answer at the end of the talk as to who's who. And they're all connected by gene flow also. So quite a complicated situation. Now, of course, the fact that there are cryptic species and hybrids, this has been long recognized, and there have been numerous influential review papers. But the earlier review papers in particular were really limited in their inferences because when you have very few genetic markers, it's very difficult, if not impossible, to make firm calls in terms of what is a distinct genetic group or a species and when is there gene flow connecting them. And so right now we're in this really great and exciting position that we have access to genomic data and we can actually accurately start to delineate who's who. And so genomics is really a game changer, and that's what I'm going to be focusing on today. So why should we care about cryptic species? I'm going to take you through a little bit of a thought experiment that I think illustrates some of the issues. So let's imagine that we are interested in temperature in corals, for example, and that we go out and we measure survival or some other fitness measurement for a group of corals, and we come up with some nice data set that looks like this, and we assume that this is one species. We will then conclude that this particular species can survive a wide range of temperatures. If we then go and genotype these individuals, and it actually turns out that we have 2 distinct sets of species in our experiment, then we would have quite a different conclusion, which that each species can survive a narrower range of temperatures. Okay? So taking this, I just want to point out, this can be any aspect. I've shown temperature, but this could be any aspect of the niche. This could be microhabitats. So it may be, in fact, that we have different distributions of these cryptic taxa living close to each other on a single reef. And so I'm just going to highlight, from a restoration point of view, a couple of examples of how this can play out, but we go through a lot more examples in our preprint, and I'll put this link online at the end of my talk as well. So for example, it may be that these different taxa inhabit slightly different environments. And so you could, if you were deploying corals on a reef, you could have mismatches between certain taxa and their environment. So these green corals do better on the green environment, whatever that means, right? You could also have differences in competitive ability between these different taxa. We don't know, right? And then finally, reproductive isolation, by definition, is going to be happening. And so if you deploy 2 taxa next to each other, the allele effects that we've been hearing about from a number of talks could be magnified, or when you're creating broodstock in captivity, you're going to have blockages to fertilization potentially happening amongst your parents. So these are some of the reasons that we might care about cryptic species for hybridization. So how prevalent are cryptic species and hybridization? To answer this question, we dove into genomic studies that have been published. There's some details there for those of you who care about that sort of stuff. But the basic thing is that we found 51 studies representing 31 species, and this is worldwide in scope. Okay, so first I'll tell you about the cryptic species, and then I'll tell you about hybridization. All right, so how do we detect cryptic species? That in and of itself, we went through a lot of thinking in terms of how could we have repeatable procedures for making this judgment call. And so again, another little thought experiment. Imagine that we have gone to two different reefs. We'll say they're geographically separated, and we recognize that we have slightly different genetic groups within these two different reefs. Now in this sort of situation, it could be that the differences between locations are due to reproductive isolation and that we have different species, or it could just be that we have differentiation by distance. This is a common phenomenon in nature. So in this case, we really can't just look at the data, just look at genetic data and know what's going on. But if within a single location we have distinct genetic groups that co-occur, this is really strong circumstantial evidence that we have multiple species, or two species in this example. And if we have this pattern repeated in multiple geographic locations, our conclusions grow in strength, okay? And so this is really the criteria that we used, is do we see distinct genetic groups occurring in symmetry relative to dispersal ability? So this is a very conservative criteria to make the judgment call. Okay, so how common are they? I'm going to show you in a minute a rather complicated graphic, but really what I want you to pay attention to are the blue circles and the green circles. So the blue circles are just showing very strong evidence for cryptic species, and the green circles are, well, maybe, no, hard to tell, right? So sort of ambiguous. Okay, so this is our main results. This is the list of species as described by the original authors. So it encompasses a fair amount of diversity, and as you can see, the dark blue circles are as common as the green circles. The columns of ordination and model-based clustering just mean some different ways of looking at the genetics. That's not important, but what I do want to call your attention to is the final column that is reporting how many times that pattern is repeated within the study that was undertaken. Okay, so then overall, we find that conservatively, 50% of studied corals to date appear to comprise multiple cryptic taxa. And many of those are more than two, incidentally, just by the way. Now, interestingly, many of these cryptic taxa segregate with depth, and I'm going to give you a list of some of those that do. They don't seem to differ by reproductive mode, which is quite interesting observation. However, only 60% of studies have reported their sampling depth, so we don't know where their corals came from in the genetic studies. Only 20% have replicated their depth sampling locations, and only 4% recorded depth by colony. And so for 96% of genetic studies, we have no idea where individual colonies are living on the reef, and that's bad. It's really bad. Okay, so cryptic species happen. What about hybridization? I'm going to show you a couple of ways that we were able to look at hybridization. First, we thought, oh, okay, let's go see which studies actually test for hybridization. And sort of shockingly to us, out of 51 studies, only 2 ran tests. And in those studies, they found first and second generation hybrid individuals. So, okay, but only 2 out of 51, so not so helpful. So we thought, okay, we'll take an eyeball, quick and dirty approach. This is a presentation of data that's really common in genetic studies. Each bar is an individual, and then we're looking at the proportion of the genome that belongs to a different genetic groups. So in this case, we have 2 very clearly defined species. This is real data, incidentally, it's not made up. And these are likely hybrid individuals, okay? So we can do this with all the studies that were published. We went through, and out of 34 studies that we could do this assessment for, 21% showed, or 21 showed patterns consistent with hybridization. So again, indicative, not the best test. And then we can also look at historical patterns of gene flow. So there are methods for estimating gene flow on a historical time scale, and basically all of the studies that used a demographic inference approach to look at historical gene flow found support for gene flow between cryptic taxa. Okay? Again, reproductive mode doesn't matter. So this idea that we have cryptic species and we have low levels of gene flow connecting species, this has been around for a while, and I think it validates some concepts we have about the natural history of corals. So hybridization just does appear to be a regular feature of coral biology, which I find personally fascinating. And I just want to point out that another group recently came up with similar conclusions, looked at data in a slightly different way than us, but a really nice paper from Sarah Davies' group has looked at similar issues that you might want to look at. Okay, so given this prevalence of cryptic taxa, are coral biologists designing their experiments with this in mind? Okay, so keeping in mind the thought experiment I showed you earlier, to see whether this is an issue or not, we turn to a data set that was pulled together as part of another study. This is just a data set of experimental studies in corals. And so we just looked through this data set and found that only 8% of experimental studies of heat stress on corals genotyped the host, and only 4% did the analyses that would allow cryptic species to be recognized. So probably about half of these studies have cryptic taxa in them without realizing it. Okay, so what can we do? So best practice that we are recommending is expect your samples to contain multiple species, record depth and other information. We're working with the EcoWrap group, so we know exactly where our corals colonies are, which is amazing. Take pictures. Russell can give you lots of tips on how to properly take pictures of your corals. For non-geneticists, consider taking samples and preserving tissues and sharing them with your genetics friends. And then for geneticists, do bioinformatics with cryptics and hybrids in mind and make friends. The environment matters. Everybody should be publishing their metadata at the colony level. And so, to wrap up, according to Google Scholar, there are 1.6 million studies on corals, and we still don't have a firm grasp on who mates with whom. And I find that completely shocking, and we can do better. We have the tools now to do better, but we need to work together in order to make that happen. So here are the answers in terms of who's who in Stellafra Epistolata on the GBR, and I just want to thank my co-authors and people who have given some really thoughtful insight into our work. Thank you.
Thanks, Cynthia. That was super eye-opening. And we have a few minutes for questions.
Do you work all around the world or in a specific geographical region?
I work here now, so empirical studies from my group are from here, but it's fun to survey worldwide data.
So I did my research in Puerto Rico in the late 90s, no, the early 90s, and I found that there really are two species. It looks like two species of Acropora cervicornis, the foundational species that they're working on, two completely morphometric. So I did that. I grew them side by side. They maintained their morphometrics, but there was anyway. I would like to share that with whoever would be interested, because I have the morphometric data on how growing side by side for over a year they maintain their difference. One of them has indeterminate growth and grows huge and has huge stems and likes reef fronts, and one of them never feeds their base and has senescence, and it's very, very different.
Yeah.
And nobody's recognized it. It's weird after all these
well,
years.
I think we need to do multi-generational studies on corals. I mean, so some of these, morphologies could be plastic effects that, or maybe persist through the lifespan, but I think this needs to be looked into more, but my knowledge of coral morphology is very, very limited. So put that out there.
We should tie that to genetics.
Yeah.
Great talk, Cynthia. Given the fact that it will take some time for all your recommendations to be uptaken, and we have to still interpret what we already have in the literature, right? I want to know how you would interpret two scenarios. There is a trait we study, and it seemed quite consistent within a species. And we say, we know what that species does. And according to what you are saying, it's very likely that in many of the samples, there was more than one, right? So should we think that means that probably the trait is strong enough that even across species probably the same? Or should we think that if we were deeper, we may split that trait into two, and then we are kidding ourselves all the way through,
Yeah,
wrong?
so, I mean, Juan, I think it is a really hard question to answer, because what it really, it depends on the proportions of different entities that you have in your data, right? So if in one pool, you have a mixture of two things, that's gonna cause one data pattern to emerge, whereas in another instance, you have one represented, and then in another instance, you have a different one represented. You're gonna interpret your data in different ways, and so I think it's a really hard thing, and I'm not willing to give you a generic answer, so I'm not answering your question.
It's okay. You can argue it more over lunch.
Yeah.
Let's thank.